All right, next up we have Erin Horkan. She is attending University of Washington and majoring in oceanography and Spanish with a minor in marine biology. She has been working here in Newport with the Coastal Biodiversity Risk Analysis tool with the EPA this summer. And one of her goals this summer was to put her feet in the ocean at least 5 days a week, which I don't know if you've managed that. Maybe you thought you were living a little closer to the ocean, but
It was pretty rainy some days.
that's basically, yeah, the water.
Alright. So, hello everybody. So as Haley said, I'm Erin Horkan, and I interned with the EPA this summer, and I worked on the Coastal Biodiversity Risk Analysis tool, or CBRAT. So first off, what is CBRAT? CBRAT is a website, and it functions as an eco-informatics platform. So at the moment, there's a public version available that has taxonomic, trophic, habitat, range, all sorts of data for over 400 coastal species. So you can see on this map here, the 13 ecoregions we're looking at, we're not including Hawaii, we're going from the Gulf of California up to the Beaufort Sea. So ultimately, the goal of CBRAT is to be used as a climate change risk assessment tool. So we're looking at the risk that sea level rise, ocean acidification, and increasing air and sea temperatures pose to these species. So my research this summer focused on ocean acidification. So all of the CO2 humans are emitting into the atmosphere, the ocean absorbs about a quarter to a third of that, depending on who you ask. And then through several reactions with seawater, I've included a Jerome plot here to show this is the pH range right now, about 8.1, and then we're moving lower. And as pH decreases, the CO2 increases, and the concentration of carbonate decreases. And so the three components of ocean acidification are important for marine organisms, and that's the CO2 concentration of the seawater, the pH, and the saturation state of calcium carbonate, particularly aragonite, the more soluble form that a lot of species larval forms prefer to calcify. So I started out looking at decapods. So I looked at over 30 manipulative experimental studies. I put kind of all of that information into a big spreadsheet, and I was identifying trends and looking for a correlation between specific pH values and a strong negative effect on all species across all studies. And then we got together with the rest of the CBRAT group, and we established these 3 cut points last week. And these are all preliminary. We haven't kind of reviewed them, we're still working on them, but ultimately we think about a pH of a 7.9 poses a low risk. It's unlikely to have a negative effect on most species. 7.7 is our moderate risk value, so it's likely to start having a negative effect on some species, and a 7.5 is a high risk. And we think that's very likely to show a negative effect on most species within this taxa. So then after I was looking at this data for decapods, I then went and looked at existing data sets to try and figure out what pH values Pacific Coast organisms are currently experiencing. And I focused on the NOAA West Coast Ocean Acidification Cruise data from 2011-2013, and then we took averages of values from 0 to 30 meters in the shallow water and found that the average pH is 7.8, and then we found for deep water from 30 to 200 meters it's about 7.5. And this data was taken in August and September, so this is when you would see kind of the strongest upwelling and probably the lowest pH values. So comparing that to our cut points, we see that species are either might already be suffering, or some species may have adaptations that were revealed in the study that I looked at that are allowing them to withstand these low pH values. So these cut points aren't necessarily as kind of cut and dry as they may seem. One of the things I also looked at was trying to find species characteristics that might make a species less vulnerable to ocean acidification. And one of the most useful papers I found was a review by Whiteley that looked at did another review of decapod data and found that species that live in high energy environments like estuaries and on the coast are good osmoregulators, have high high metabolism and are more active are less vulnerable to ocean acidification. So then that leaves the more vulnerable species would be those typically in like polar latitudes and the deep sea where they've experienced less environmental change over their history. So then how would we use this data in SeaBrat is to downgrade a species risk based on those characteristics. So from where I have Dungeness crab and then tanner crabs, a study looked at how immersing these two species in really low pH conditions and then measuring their responses and their ability to recover. And they found that Dungeness crabs, the typically estuarine species, was able to recover much faster. And so that's good evidence to use that in SeaBrat. And then if we were looking at these two species in a specific ecoregion that's expected to have the same amount of pH change, we would then downgrade the risk of the estuarine species. So that's what we're working on right now, and I've compiled similar information for fish, and so we're hoping to develop cut points for rockfish next week, maybe, and continuing research to look at kind of differences in the response of larval and adult organisms, and then the effective combined factors, so like temperature and ocean acidification together seem to have a combined effect and be even more detrimental. So I learned a lot this summer. I'd really like to thank my mentors, Henry and Christina, and my fellow office mate, Maya, as well as all of the other Sea Grant scholars and Oregon Sea Grant for making this possible. So thank you. So any questions? Yes.
So there's some research that indicates that saturation state is a bigger driver than pH. Are you also looking at that, or are you primarily just looking at pH for these species and others?
For decapods, I found that the studies primarily looked at pH and PCO2. Decapods use kind of chitin and calcite more in their exoskeleton than aragonite, but it does seem for bivalves, aragonite might be a critical factor. So for decapods, we were started to look at maybe developing aragonite cut points, but we didn't get to that yet because the spread of aragonite values across studies was really varied, and that wasn't the main thing they were controlling for. But I think for bivalves and maybe echinoderms, that might be a bigger factor.
Do decapods use aragonite?
Some do in their internal part of their shell, yeah.
Well, then that would be controlled by internal chemistry though, wouldn't it?
Right.
So the aragonite saturation state shouldn't be an issue. How far apart are the aragonite and calcite saturation states?
That I'm not totally sure about. I was only looking at aragonite data, and most days you have one or the other. But that'd be something to look into. Generally, aragonite's more soluble, so that's the one.
I haven't heard people talk about calcite saturation state yet.
Yeah, it's generally only in terms of like the CCD, not necessarily in terms of organisms. Yes.
So, what scale did you use for the pH? Because pH is measured on a bunch of scales. That's one of the issues with pH. Did you make sure that they're all on the same scales?
I didn't look too closely at that across the different studies. I read through kind of the methods and what they reported as their target pH values for what I put into the table, but that would be something to look at. Yeah.
Well, somewhat of a follow-up, in your post you mentioned about the variability among species. So how much of that variability is not species, but the experimentation? Because the experiments seem to be all over the place in terms of how they're done, the endpoints, and maybe even how they measure, well, certainly how they measure pH. So can you have any idea of the biotic variation versus the experimental variation?
The experimental variation was kind of generally the variation in what response variables they were looking at. So all the studies I looked at, it was some were looking at mortality, some were looking at just physiological, like swimming activity, that sort of thing. And then within species, they would directly compare kind of like what, like for this example, what kind of habitat they're found in, and that seems to be the main one in between species is if they're more active in a more active environment versus in the deep sea. So I'm not totally sure. seems like the more important thing is the species variability. The studies are all testing kind of low pH, high PCO2 environments, but they just look at different responses. There was a lot of variation in the response of larvae versus adults though, and that is kind of reflected in our cut points. One of the things I think why there's the deep water pH is so low, and that is our high risk, is that a lot of decapods, the larvae spend time up in the water column, so it's possible that this high risk is more targeted towards larvae than adults. All right, thank you.